Okay, so we can get started. So today I'm going to talk some more about the co-splicing networks and how is it different from the co-expression and the co-splicex networks. So we've seen that you can construct networks and put them into kind of discovery mode. But there's an alternative to that, which is to go more into kind of hypothesis-driven research. And I think that if you do network analysis, you should do both. So what did I want to accomplish in this project? So I constructed this novel network, and I wanted to show that it's different from what has been done before. So for that one, it's better to go kind of construct hypothesis, and say, yes, this is the factors that affect this particular type of network versus the other one. So that's, to me, that falls into the category of kind of hypothesis-driven research. So the first question I asked is, what generates all these network edges that we're constructing networks? And all we have is a matrix of correlations. So now the question is, what generates this? So what we want at the end of the day is to understand the biology. So for that, you can go to, like I said, in a hypothesis-driven mode and say, OK, my hypothesis is that protein-protein interactions will give me strong edges. So for that, you go to databases, of which there are many, and you download all the known gene-gene interactions. You overlay them on your networks, and you say, OK, I want to compare the edges that are between the known protein-protein interacting genes versus similar groups of genes that are randomly selected. So here's the outline. You collect all known protein interactions, overlay with the network, and so on. So here's what we find. So I have two sets of edges. Some of them are between PPI genes, and some of them are between random groups of genes. I compared them using the Wilcoxon test. And what I plot here are the confidence intervals. So if the confidence interval is above 0, then I have stronger edges than expected. And if it's overlaying the 0 line, then I don't have evidence for that particular factor. So what we can see is for all types of networks, for the co-expression, all the genes that have known protein-protein interactions are co-expressed. They're strongly co-spliced. And also the co-splicex, which is a combination of co-expression and co-splicing, also I have strong edges on there. So my hypothesis is validated. If you have if two genes interact together, presumably they are regulated together, so that you should detect that at the level of the co-expression. Okay, so now how about transcription factors? So I go to the database, and I download for each gene all the known transcription factor binding sites. I do that for every single gene. So now I compare. I have for gene 1, I have 5 transcription factors, let's say. And for the other gene, I have 4 of them. So how do I compare those two sets? So if you have, in this particular case, I use the Jacquard measure, which is the intersection divided by the union. So how many do they have in common? So I have C, D. I think only C, D is in common. And I have, I think, 6. So I have 2 over 6. That's the measure, Jacquard measure in this case. Okay? So I collect all edges that the Jacquard similarity is more than half. So the intersection over the union is at least half. And I compare that with random groups of edges of the same size. So what do I find? As expected, the co-expression network will have strong edges based on transcription factor binding site sharing. but not co-splicing, which is also to be expected because transcription factors generally shouldn't affect splicing that much. And the co-splice X is affected because it's kind of a hybrid. It's sensitive to co-expression and also to co-splicing. Now, splicing factor binding sites. So these are in both introns and exons, and there are a lot of them. And also, they have high multiplicity. So the same kind of degenerate binding site will be present many, many times within an intron or an exon. So the question now, how do I compare? So for each gene, I download all these splicing factor binding sites. Actually, these are computationally predicted. And I have two sets now of like each identity and the multiplicity for each gene. So these are the splicing factors that I considered for this, that are available in this database. So I have 21 of them. So for each gene, how do I represent this information? I have a vector of length 21. And for each entry in the vector, I say, OK, what's your multiplicity? So now for another gene, I'll have another vector of length 21. And what is the multiplicity and identity of all these splicing factor binding sites? So for this one, I use Canberra metric. You can use other distance measures. I don't think it's essential you use this particular one. You just have to understand what the information is and make sure that the distance measure captures that information. So I choose the distance in the bottom quantile. So basically, they are similar in terms of which binding sites are present in each gene. So what do I find? If you share splicing factor binding sites, you're not going to be more co-expressed than expected by random. But you will be co-spliced, and you'll also have the co-splice X interaction will show up there. So this is exactly as expected. So kind of, I feel justified to say that I have a co-splicing network because it's sensitive to splicing factor binding sites and so on. MicroRNA. So microRNA, they bind to genes, and they kind of interfere with transcription. But there's another mechanisms that it's presumed to operate here, which is that you can attach to a transcript or interfere with the transcript and just make it less stable. Okay? So the first mechanism would explain why you're more co-expressed. If you knock down the same genes, then these genes will be co-expressed. But why would I have like lower co-splicing? So that to me, that means that if you interfere with the transcript stability, then it's going to be more noise in how many introns and exons you are working with. So the data will be more noisy than for genes that are not the targets of microRNAs. So therefore, they will just show up as uncoordinated. And the same for co-splice X, because remember, this is sensitive to both, so it will pick up the interference with the splicing. Chromosomal folding domains. So genes are on chromosomes, and the chromosomes fold. And then when transcription happens, some of these folds get exposed to the cytoplasm, and transcription starts to happen. So the idea is that if genes are close on the same folding domain, that is likely that they will be expressed at the same time. So what do I compare here? So I compare genes that are on the same folding domains versus the genes that are at the same base pair distance, but not on the same folding domain. Because if you compare genes that are close by on the same chromosome versus genes that are on different chromosomes, that wouldn't be really a fair comparison. So this is a fairly strong result. However, you're not more likely to be co-spliced or co-splice X. So that means that if you're not co-spliced for other reasons, just being on the same folding domain will not increase the co-splicing. So previously working with just microarrays, I found that if you look at the mouse Allen Brain Atlas, you find that genes have like spotty kind of expression, even in the striatum, which striatum is considered kind of a more uniform, among the more uniform brain regions. But expression of the genes is spotty if you do whatever imaging they did here. So genes are not uniformly distributed. So now if you take two genes and you overlay and you say, okay, are your spots overlaying on the other gene's spots? And you do a spatial correlations. So the Allen Brain Atlas has a tool, it's called Neuroblast. So for each gene, you put the name of the gene and you say, I want the striatum and I want the genes that are kind of spatially co-expressed with these genes. And it gives you 250 genes that are the closest ones. So I collect all the 250 edges. And for all genes, I repeat this process. So then I have a set of edges between genes that are spatially overlapping. And I compare them with genes that are not likely to be, not as likely to be spatially overlapping. So what I find is that it's a strong co-expression and not very strong splicing. I think I have an error here because actually the splicing is very strong. So I think this is the transcription factor image. So please check the website because I'll put the correct image on there. Okay, so now I will look again at the top co-expression and co-splicing hubs and say, okay, are there strong edges between genes that are just like kind of co-expression hubs and co-splicing hubs? So remember, a gene can vary just its expression level and will not have many isoforms. So then the question is like, can it be kind of related with some gene that has a lot of isoforms? So to address that question, I went and I found, okay, the top co-expression hubs that are not co-splicing hubs, and vice versa. So I find the top co-splicing hubs that are not co-expression. And I have three types of networks. So I say, which network will give me strong edges between those types of genes? Okay, so for each network, I collect the edges between these different types of hubs, and I look at their quantile distribution. So if you look at the co-expression, it has very weak edges between these hubs, the different types of hubs. And the same is true for the co-splicing. It doesn't have any strong edges between top co-expression and top co-splicing hubs. However, my favorite network here, it does find those strong edges. So in conclusion, the co-splice X, which, as you remember, it's constructed using the Canberra distance, is the only one that finds coupling between co-expression and co-splicing hubs. So if you have a splicing factor and it doesn't have many isoforms, you would expect it to go and affect the splicing of the genes. And if you look just in one type of network, in the co-expression or the co-splicing, you will never find that, even though it's very meaningful biological interaction. Okay, so we'll switch gear a little bit over here. So all the results I've shown you are from a data of 60 mice, RNA-seq from the striatum. But in reality, there are two categories of these. So we started with a genetically diverse population of mice, and we selectively bred, so for a behavioral phenotype. So the behavior is you inject them with an antipsychotic drug, haloperidol, and they either go cataleptic or they do not. So the purpose of this exercise is to find what are the genetics that make people have these terrible side effects from haloperidol. So we want to find what determines, catalepsy in this instance. So I have 30 highs and 30 lows. So then I constructed two networks, one from the high and one from the low, and I look at the connectivity distribution. So what you see there is that the highs have like much stronger connectivity than the lows, and in the co-splicing network and the co-splicex, it's even more striking. So what is going on here? Why do we, and I'm sure that some of you have found this when comparing two networks. So remember that our variability is genetic. So we don't have like time series data like we've seen in the previous talk. So all the variability is genetic. We sacrifice the animals at the same time, so there's no confound there. So to understand this, we'll have to take a brief foray into mouse genetics. So the B6 and all the other inbred lines are basically clones of each other. They have pretty much the same genome. And they look the same as you would expect. And there was a problem with that because humans are not genetic clones of each other. So people have developed a population of mice that is genetically diverse. So now the genome of these mice, it's all different from each other, and it's a mosaic of these original inbreds. So that's the population that we started our selection from. And now we have our high group and low group. And here is just an example of some other fun phenotypes that we are working with. My collaborators breed them for preference for alcohol. So some of the mice, after a few generations of breeding, they'll really drink a lot. And the more generations you breed them, the more they will drink. Okay. So all these effects that I'm showing you for the haloperidol group, they're even stronger in these alcohol-sensitive mice. Because these ones, they were bred for alcohol, and they went for 20 generations possibly. But the haloperidol, we just went for 3 generations. Okay, so back to distance measures now. So you have 2 genomes, and you genotype these animals. So you have a whole bunch of genotype data. You compute the genetic differences. So the way I do it is the simplest possible way. I just encode each allele 0, 1, or 2 and compute the distance between them. So you end up with a genetic differences matrix. So you can observe that you can include as many markers as you want in here. So you can include just one genotype, one marker, or you can include 2. You can include a chromosome, or you can include the whole genome. So for now, for each pair of markers, I compute the genetic distance. So now I have for all the markers on my genotype ring array, I have this square matrix. So what can I do with this? So let's suppose that I want to separate these animals, and I want to say, OK, these are the loci that will discriminate between these 2 populations. And let's say that I also want to find some sense of optimal set of these markers. So instead of just going and collecting a set of p-values and doing an FDR and then setting an arbitrary threshold for the NBR, for the FDR, I say, no, I want something that will really maybe be biomarkers. Okay? So I want to find the best out of all my markers on the genotyping array, what gives me the best discrimination power. So the algorithm is the following, and it's really based on work by Nicholas Schork. You can see the paper here, and I highly recommend it. You correlate each marker distance with the phenotype. So my phenotype is just 0, 1. Are you high or are you low? Order the intervals in decreasing order of significance, and then compute the distance based on the first most significant markers, the first two most significant markers, and keep adding ones, and then computing the correlation. So here we go. Now we have our ranked markers, significance. So what happens is like the top correlation is 0.23. The second one is 0.2. Together, though, they give me higher. And then as you add more and more, you eventually reach a peak. And then adding more markers actually decreases performance. So this is a kind of very easy example. So this doesn't happen all the time. So sometimes you go high with a few markers, and then you go low, and then you keep increasing again. So my interpretation of this is that you have two groups of genotypes, one that are like really strongly associated, and then you have a second larger group that have just like these small infinitesimal effects, and they can coexist. So sometimes adding those small effect markers all together will take you higher than the first peak, and sometimes it will stay lower. So, yeah?
Just wanted to really understand the procedure. So let's say I want to find markers for body weight in mice.
Yes.
So then I run my genome scan and I get SNP marker and I can rank them by p-value.
Yes.
And so then, okay, so then you first focus on the number one hit.
Yes.
Distances between the mice.
Mm-hmm.
Distances between the mice according to body weight correlated. And then you see here the
Yes.
R measure. So
Exactly.
and then you add in more and more markers until you have this leveling off effect.
Yes. So that's exactly it. And I'll show you later that you can do this with expression or with splicing data, really any type of data. And I should also say that I do not claim that this is a global maximum, because I can do better. So instead of, I start with my top marker, and then instead of adding the second one, I try everything else. And then I keep doing that, so just have lots of coffee and let the computer run, and then you find improved performance. Okay, and then you can do even better, because you can take that initial point and you take it into a genetic algorithm, for example, and you give it, this is your starting point, and can you find anything better? So that improves it even more.
Is this similar to doing a random forest? In what way is it better?
I didn't compare it.
I mean, I would say it's quite different from a random forest. In a random forest, you would have let's say the outcome is mouse body weight, and you use all the markers at the same time.
Right.
But that's really.
So what's the kind of modeling you use? You use a regression form to predict the rate, or
I'm sorry?
like when you keep adding,
Mm-hmm.
when you say adding to predict the significance,
Yeah.
are you really using a regression model to check the significance, or?
So what I'm plotting here is the significance, which is the correlation between the matrices. So I have a matrix of body weight, which is 60 by 60, right? And then I have a matrix for each marker, 60 by 60 again. So now I can just add the first two matrices. So then I have another two matrices, right? The combined genotype, and then the.
You add each set to each other set.
I add these matrices to each other.
Matrix to the new feature you added.
Yeah.
New matrix of sample by sample you add.
So these matrices are of the same size, so I can add them together and correlate and add the sum matrix correlated with the phenotype matrix.
Okay, but the first matrix is based, for example, on marker 1. The
Yes.
second matrix involves markers 1 and 2. Did you use?
Yeah, so here the first matrix is the first marker, the second matrix is the second marker, and then I do a sum matrix,
Yeah.
which.
Did you ever try, or did you ever try using a weighted average sum of it?
Weighted average? No, I haven't tried that, but it presumably could help. So maybe, yes?
Kind of like a force selection
Yes, forward selection, exactly. So all the details are on Nicholas Schork's paper. So it's forward selection. You can do variations on this. So you start with all your markers and then subtract and say, I don't know if that would do better or not. You know, subtract the least significant one and keep subtracting. So what are the results? So now I have my distances, I have my categories, I plot them on 2 dimensions. You can see that there's no question that I can separate my samples. Okay, a very important detail here. So these are the highs, and these are the lows. So what do you see there? First, that they are separated as I wanted, but also that there's more distance between these guys. So now they are more far away in terms of genetic distance, but the lows are closer together. So that means that they share more genotypes in common. Okay, so what happens when you breed them? So when you breed them, you know, there's you have a new baby, right? So now the genome, there's linkage there. So the genome kind of like rearranges, and then that gives rise to genetic linkage. But this is more complex now because you also have genetic drift where some alleles that are rare disappear. So this doesn't happen at the same speed for some reason in the 2 selections, for the highs and for the lows. So what you end up with are some mice that are kind of, for lack of a better word, kind of inbred together a little more. And these ones, for some reason, they are a little further apart. So I will show you that this generates effects on the networks. Okay, so now I just presented you with a different way to analyze this data. So you might ask, okay, how does it compare with previous methods like EdgeR? Let's say that I take EdgeR and I do differential expression, and I have a set of p-values. Now I do my own method, and I have another set of p-values, and I say, how concordant am I? So some of you might be familiar with the area under the curve. So what that does, you select like a threshold, and you say, EdgeR, you're the gold standard. Whatever you give me under the p0.01, I consider the truth. And whatever my method doesn't find at that threshold, it's error. So you change the threshold, you vary this p-value threshold, and you find the true positives and the false positives, and you plot them. So the idea is that if you have a curve that reaches close to that point, and you measure the area under that curve, if you're close to 1, you're doing really well. If you're not doing that well, then the curve is going to look kind of like that. And if you're doing kind of random, then it's going to be close to the diagonal. Any questions about this? So basically, my Manhattan distance measure comes very close to results from EdgeR. However, if I go and use the Canberra distance measure, that will be sensitive to splicing. So it will find me significant results that are really due to splicing. EdgeR doesn't find that. So that's why the performance is lower using that distance measure. Okay. Now the same idea, instead of using genotype data, you use either expression data or splicing data. So correlate each gene distance. So you have a gene, has a bunch of expression levels. You compute the distances based on that, like we talked yesterday. And you correlate that with your phenotype, and you find significance. Keep adding significant matrices. So what is the performance? So as you see, eventually you reach a peak, and then you start going down. Same story. And for expression, if you just look at the expression level, then you don't do as well. So basically, if you add more exons and you just select the significant exons, then you're going to have better discrimination. And here is just a two-dimensional projection of these best distances I found. So what are these colored dots over here? So I said, okay, so now I have this co-splicing network. And I don't know if you can, I was trying to find some kind of like module significance kind of measure. And I was not satisfied with just putting all the exons and doing eigengene on the exon level data. So I said, what if I restrict my forward selection to each module separately? And say, okay, the best performance you're going to get is how well the genes in this module will discriminate my samples. And I find for each module, coded by color, some level of significance. So for a while, I was excited about this. But then I thought about it, and I said, okay, so what happens if I just go and now I select random groups of genes of the same size as my module? What kind of significance do I get there? So I find that selecting random groups of genes doesn't give me, it does give me the same performance as a module. So if I have a module of size 200, it reaches some significance there. I really have to compare that to with 200 genes that are not in the same module. So I repeat that, and I compute a z-score, and none of them were significant. So that means that if you select groups of genes at random, you do as well as with any module. So it's not a good method of finding module significance. So I think in this case, I'll just go back and just use gene set enrichment. So you have the p-values from each of your module. And then you say, are they lower than what can be expected by chance by that selection? So in that case, you do find significant. Yes?
I'm sort of lost somewhere in the previous step itself. The question is, why not we directly do a differential splicing analysis or a gene level differential isoform analysis and choose those exons as the features to construct the network? Why we have to choose this kind of a marker selection approach?
The question is, if I understand correctly, why do I like this method of using matrices versus just using established tools?
Oh, no. So I do understand the matrices, but you're choosing certain set of markers, not the whole genome as a marker for doing the significance analysis, right? And how you're doing is you're doing a sort of like univariate regression for forward selection, having all these weights as equal, just adding up all these matrices to check them. But in order to select these matrices by means of the approach what you're showing now, why don't we directly do first differential exon level differential analysis, differential expression analysis, or isoform level differential expression analysis? Choose those exons as the markers for this.
You can do exactly that. And so I think in EdgeR, I just did that at the level of the genes. So you do differential expression, right? Based on each gene, you rank the gene, and then you combine the genes, right?
I was trying to understand, does this give some better performance in terms of splicing networks rather than choosing the differentially expressed exons?
No. So the reason to use this is because the forward selection. So the only reason to use this as opposed to differential exon usage, differential exon expression, is because I can combine them in this way. If you just restrict yourself to differential exon usage, how are you combining them? So the benefit on this is that you can combine them easily. Is that what you're asking?
Yeah.
Yes?
I may have missed this or it may not be relevant, but are you doing the significance testing for the group in the same, like in your training set, in your same set?
Yeah, so
overfitting?
Yeah, so the set here is fairly small. I have 30. So yeah, you're totally correct. I would have to find a bigger data set and see if I'm overfitting or not. This is just the standard way that you construct a biomarker set. So now I can construct splicing distances. I can construct genetic differences. So then there's nothing stopping us from doing splicing QTL detection. So there are several papers published on this topic. So basically, I'm just trying to show you that using distances, you can detect splicing QTL and expression QTLs. And if you do just a literature search or email me, I'll send you the relevant papers.
Could you explain this figure?
OK.
What's the pink arrow, what's the blue arrow?
Right. OK, so I have RNA-seq data, and I map it to exons. So suppose I have 3 exons. And these are the reads that map to exon 1, exon 2, exon 3 in one sample for one gene. I go to the same gene on another sample. I count the exons. And then I represent this data as a vector in 3 dimensions, or 7 dimensions if you have 7 exons. So now you have 2 vectors, and then you can compute the distances between them in several different ways. And some of the ways are sensitive to just the angle between the vectors, and some of the distances are sensitive to the magnitude of that vector. So if you're sensitive to just the angle, then it's more of a co-splicing. And if you're sensitive to the expression level of the whole gene, all the exons going up at the same time, then you will increase the amplitude of that vector, leaving the angle unchanged. Or if you just choose one exon to go up, then the angle will change, even though the magnitude might not change that much. So basically, co-splicing is based on the cosine, based on the angle, and the co-expression, the co-splice X is based on the Canberra distance, which is sensitive to both. So any questions about? So this is just a summary slide. I have Manhattan distance between the genetics. I have phenotype differences, mental correlation, and QTL detection. So if you are interested in the details of this regression procedure, here is one of the best papers, I believe. So I really highly recommend reading this.
Why is it called multivariate distance regression?
I think because they choose multiple features, so that you add together the matrices. So, there are many ways of finding splicing QTLs. So once again, why do I prefer to use distance measures? Because sometimes the data is really highly complex. So you imagine that you go and you image these 2 people or several people, okay? And then you find structural differences between brain size, or sometimes it's not even that. It's the folding. Imagine that you detect differences in the folding of the cortex between these people. And it doesn't correlate with the size of the thickness. It's just really subtle patterns in the folding of these regions, okay? So what are you to do with that? What are you going to regress on? Okay, so one of the things you can do is you use a deformation distance. So you say, I have this cortex from this person. I have this other cortex from this other person. What is going to cost me to deform one cortex into another? And that's your notion of distance. Sometimes you go and you kind of measure really complex behavioral phenotypes, for instance, impulsivity. So you go and you say, OK, I'll give you $5 now or $50 tomorrow. So one of the findings is that people that use meth or other kinds of fun substances, they tend to become very impulsive. And that's measured as a curve or even as a surface. So now you have 2 surfaces for 2 people. So what you can go and you say, OK, I'm just going to choose some kind of measurement, kind of a brute force approach. I'm just going to choose this because I need something to regress on. But you can be more, I believe, sophisticated about it. And you go and say, OK, I'm going to compare 2 surfaces or 2 curves. and I'm going to construct my distance based on the difference between those complex measurements. Let's say you go and you find like functional networks. So you have like voxels in the brain, and they talk to each other, and you construct a big, huge network. And then you look at two subjects, and they have different networks. And you can ask the question, is that genetically determined? So you can do this. You can just construct your distances. Then you have your Mantel correlations, and then voila, there it comes. The problem with this approach, though, is that you're throwing together like a very large amount of data. So that's why I talked about like selection, feature selection, the forward selection. Because sometimes really the differences are localized somewhere. So then what you would do, going back to the previous slides, you take your strongest little feature, and then you say, okay, this is the most related to the genetics. And then you keep adding, and until you find a peak. And then you say, okay, these are the features of the functional networks that are really most related to the genetics. Okay, so back to our networks. So remember, we were puzzled by this difference in connectivity, especially in the splicing connectivity. So what is the cause of that? So remember, we have like many genetic markers, and you can construct a distance for each one of them. So now you end up with like all these distances. So it occurred to me, like, why not put them in a network? So what does that imply, though? That implies that there are correlations, not just in neighboring markers because of linkage, but there's some complex genetic structure in this population. So some markers on one chromosome will be correlated with some other markers on the other chromosome. So I constructed my genetic correlation adjacency matrix, and it comes close to scale-free, about 0.65. But that doesn't really matter. So remember, we have this genetic variability, which we observe to be higher in one of the populations versus the other. So now here are my genetic correlation matrix values. So I compute the connectivity for each marker in the high and in the low. And you can see that there's stronger connectivity, so stronger linkage and stronger correlations because of how the selection is not symmetric. And the expression and the splicing follow from that. So what does it mean for human data? So if you are going to compare populations with different amounts of genetic variability, so there are some isolated populations that everybody's cousin with everybody. They're going to be very close. There's going to be a whole lot of correlation, linkage, and also cross-chromosomal correlations. And that will reflect in co-expression and co-splicing networks. So I believe we have some more time, more on distance measures. So I started all of this kind of distance measure-based approach when I was a graduate student. So I was working in a lab that analyzed like spike trains. So you have a neuron, you present it with a stimulus, and out comes a spike train. And I found myself like really dissatisfied with how we analyze this data. It's like some people believe that it's the timing of each spike. Sometimes some people believe that it's the rate, and so on. So it was nothing rigorous about it. So then I came upon a paper that said, okay, spike train metrics. So you compare how close is this object versus this other object. And it's based on the idea of a cost-based matrix. So, okay, you have like a spike here that's not present here. So I'm going to add a spike there for a cost of 1. And also, like if the timing is different, then I'm going to adjust the times a little bit, and every single step is going to cost me something. At the end of the day, I find the minimum cost that will turn this object into that object, and that's my distance measure. Okay? And you can adjust how sensitive you are to timing or how sensitive you are to rate. And see where the information is, because you can see, okay, if I choose a distance measure that's sensitive to timing, then I have this much of a explanatory power versus if I choose some other distance measure. So this is just the outline of the procedure, how we compute the distances. So the take-home message here is that much of the data that we are working with is really highly complex. And sometimes we just choose like features because it's convenient to regress on them. So we don't have to do that. We can just be more integrative about it. So another project that I worked on as a graduate student was analyzing songs from the zebra finches. So this one has this one song, this one has another song. How close are they together? Okay, and now let's say you go and you kind of do transcriptional analysis. Is there any relationship between genes that get expressed in the brain and what kind of songs they sing? So basically, almost any kind of data you can analyze with this method, and I don't think you lose any power of detection, and actually you can do better. And you know, when I find myself in lack of inspiration, I just go to the ecological literature, and they always inspire me. Okay, so that's all I have, and I think we have plenty of time for questions, and I have some code examples that I will go with over